Good afternoon. It's my pleasure to welcome faculty, students, guests, family members to this, what will be a very fun event. Really recognizing MG for all his great contributions. As many of you know, MG received his bachelor's from Caltech and a PhD from MIT in chemistry and inorganic chemistry. I thought always that was very dry, but you did a great job. And then he did an NIH postdoc. I'm an engineer, so I can make fun of the chemists. And then did an NIH postdoc at Stanford. He then went to UVA, where he was an assistant professor and then promoted to associate professor. And then he moved to the Scripps Institute in San Diego and was there from 98 until 2013, where we had the great fortune to recruit him to Georgia Tech, where he's full professor in chemistry and biology, and he's also the chair of chemistry and biochemistry and the director of the Pediatric Technology Center, and he holds the James A. Carlos Family Chair for Pediatric Technology. MG is really an exceptional and phenomenal scientist. He's made huge contributions. He's going to be talking today about click chemistry, which, as many of you know, was the Nobel Prize this year. And MG made huge contributions, particularly in his collaboration with Barry Sharpless. So we'll get some of those insights in that. I also want to add that in addition of being really an exceptional scientist, he's a great mentor to his group. And he's a fantastic colleague, and I always enjoy his really, great humor, humility, and generosity. And I think we all feel that we're very fortunate to have MG as our colleague. So, MG, looking forward to your talk, and thank you for agreeing to doing this.
Thank you, Andres. I really am extraordinarily grateful for the chance to talk about this subject, to be the recipient of this event. It feels like, I'm at a wake. I'm at my own wake. This is great. But I'm really going to give you a chemistry talk today. Not too much chemistry, I hope, for those of you who aren't chemists. But this is going to be about the Nobel Prize this year in chemistry. So you're going to have to suffer through some of that. But what I hope to convey is why this subject got the Nobel Prize, why it's important, what it's all about, and how it might actually influence and impact and help a lot of research that is being done by other people in the room who aren't necessarily chemists. The whole point of click chemistry is really to democratize chemistry. Now, I'm going to be pointing here. I think you can see this for the folks online. So we're really talking about three marvelous scientists, and I'll tell you a little bit about them. But I'm going to tell you about what this is, and then talk about our work in this area as well up to the present day. Carolyn Bertozzi, Morton Meldahl, and Barry Sharpless shared the prize for the development of click chemistry and bio-orthogonal chemistry, and these are, of course, closely related. I will be telling you mostly about Sharpless today because he was the one who formalized and really developed the concept of click chemistry. I will touch too briefly on Carolyn Bertozzi's work. I won't have time simply to go over all of it and its impact. And I will give the shortest shrift of all to Morton Meldahl, not because Morton doesn't deserve much more credit, but I only have a certain amount of time. And you will see where Morton's group contributed. But again, my apologies for not being able to cover the entire breadth of the subject. So chemistry, as I write here, is an architectural endeavor. This is something that non-chemists don't appreciate. We build stuff as chemists, and we build stuff that has three-dimensional structure. And structure determines function, right? So here's architectural things you can see. But the things that chemists build, we cannot view directly, right? We see leaves, these are beautiful. We do not necessarily see, we cannot see the molecules that make that color. But to a chemist, to an architect of molecules, these are quite lovely structures. They are beautiful in and of themselves, just as architecture is beautiful. However, most of us, of course, don't get a chance to look at structures as three-dimensional objects. Now, a few of us do. Those of us who happen to be lucky enough to do X-ray crystallography or cryo-electron microscopy or use the results of those techniques can actually perceive what these molecules are. And to a chemist, the three-dimensional shape, the effort that it goes into creating that, to constructing it in the laboratory, is really an exercise of both beauty and great skill. The static structure of this molecule is up there to the right. This is Taxol. Most of us, however, can't appreciate the three-dimensional structure because we don't have an opportunity to look at it and play with models, as I do frequently in my office. But instead, of course, chemistry is valued for what it does, right? So this is Taxol. This is a medication. You will, if you are unfortunate to have late-stage cancer, this is one of the medicines you will probably get. And so chemistry is really all about producing useful function. And that's the key message of click chemistry. So we're going to dip in and out of chemistry here into personal and then chemistry. So it's the early 1980s, take you back, whatever your favorite 80s song might be. And there I am sitting in the corner there with hair, right? And this is an undergraduate who was working with me at the time in the laboratory of Professor K. Barry Sharpless. This is Barry around that time holding up models of two three-dimensional structures that are opposite handednesses of each other, chiral structures. And this indeed, the development of the first man-made catalysts to be able to make such structures independently was why he won the Nobel Prize in 2001. This person here he'll recur. He was an undergrad. He's actually quite important in this story. My hair went the way of everything, but then we're going to skip a couple of decades to the 2000s. So there I was as a graduate student. Now here I'm a faculty member at Scripps. This is our first few years at Scripps, and actually this is a combined group picture of a few people in my lab and a few people in Jack Johnson's lab. Jack's name will appear later, I think. And drug discovery around this age, we're talking now the late 90s, early 2000s, was really not going well. The drug discovery industry was in trouble. There was a lot of talk about why don't we have a lot of new drugs. Here's the number of new drugs approved in the US. And a lot of these are kind of repeats. So the actual number of truly new drugs was really quite small for many, many years. And you can see the uptick here. And this actually has continued to go up because of antibodies, because of biologics, not because of small molecules. And the world of organic chemistry is driven by drug development. Right? It's where a lot of the money is. It's where a lot of the jobs are. Right? So organic synthetic chemistry is really intimately connected to drug discovery. Here's orphan drugs. The number here, previous to 2000 was really criminally small. Orphan drugs means drugs that treat diseases that are so-called orphan diseases that don't have no other medication, typically are diseases of the developing world, not a lot of money in it, and organic chemistry was failing miserably at this kind of thing. So chemists in the late mid-90s, early 90s, mid-90s hit upon a new strategy. Let's, instead of making a few molecules, let's make a lot of them and see if they have function. Okay, so that's the field of combinatorial chemistry, and actually combinatorial biology, getting biology's engine into the laboratory to do this, was actually also blooming at that time. This undergraduate is John Ellman, who became professor at Yale and was one of the leaders in combinatorial chemistry, one of the earliest people to conceive and publish in this area of making a lot of molecules. I'll tell you how you do that in a minute. This is the cohort that won the Nobel in 2018 for 2 out of the 3 of them for harnessing the power of biology to make a lot of molecules quickly in order to search for function. Okay, this is the province of phage display, George Smith's invention, and Greg Winter's invention to take the antibodies developed from phage display, which is making many millions or billions of molecules, picking out the one and making it adaptable for use in humans. So if you're on any biologic now to saving your life or helping your quality of life, you can thank Greg Winter and George Smith, among many others. I won't have a chance to talk about Francis Arnold, a dear friend, who evolved enzymes for function. But what we're in the realm of here is making lots and lots and lots of molecules and then picking out the ones that have the desired property. Okay, so this is how combinatorial chemistry works. Let's say we start with 3 molecules, we put them in a flask, we run 3 different reactions. essentially we run the same reaction 3 times with 3 different building blocks. So then we take these 9 components, 9 products, we combine them all, divide them into 3 pots, and run another reaction with each of an individual 3 components, and we keep doing that. And so here, as this little example, I've run 4 reactions, but I've obtained 81 separate products, right? This is the art of combinatorial chemistry. And this was really flourishing at the time. Okay, so, however, I should say, it was a niche market, as it were, a niche area. And organic chemists, organic synthetic chemists, the big names in the field, the ones who you looked up to and respected, they still made their bones making tough molecules, okay? Typically natural products, making the products that biology had made that have an interesting, important function, but that challenge the art of the synthetic chemist. And, you know, here are some famous examples. Typically, these are secondary metabolites, toxic compounds. Many of them get adapted and used as anti-cancer drugs or whatever. But the point here is that you weren't really seen as a good organic chemist or at the cutting edge unless you could do stuff like this. Okay, fair enough. However, okay, this problem the drug discovery problem wasn't being solved by this really high art. It's amazingly difficult, amazingly beautiful to make structures like the ones I had on the previous slide. Just the art and the science of it, deep insights into chemical reactivity, things that take my breath away. Right? I can't do that. I've never been able to do that. But the problem wasn't being solved. Drug discovery was still in the toilet. Okay, and the problem, this is a science devoted a whole issue to this, an interesting article, and basically the statement here is it's not even making a lot of molecules that's the problem. It's you got to make the ones with the right properties. We have to somehow hook synthesis to assaying properties in a faster, better, more efficient way. Okay, so that's in the zeitgeist. Again, we're still traveling. This is a paper that made a huge impression on Barry Sharpless. Published by Regina Bohacek and Wayne Guida, then at Florida, assessing what is the total possible landscape of the number of small molecules you could make that were drug relevant. Okay, so here we're talking about things less than 500 molecular weight, this was before biologics, so people thought a drug had to be a small molecule, made of common elements, has to be stable, all of that. They did this amazing calculation and came up with estimating the number of possible structures as 10 to the 63rd, which is, you know, getting near the number of molecules in the universe, right? And the number of molecules that had actually been made at that time was about 20 million. So, you know, the numbers are staggering, right? In other words, there's no way that we can make anywhere near approaching the number of possibilities for any one solution to a problem. However, biology doesn't play that game. Right? Biology makes functional molecules. The best engine we know for making functional molecules is biology by leaps and bounds. And biology does not play the combinatorial game. Right? Biology hooks small things together to make functional structures and adapts them by mixing and matching components to change properties. Right? So this is a slide from my intro to biochemistry class. Right, where we make proteins this way, or biology makes proteins this way. Amino acids get linked into proteins, right? Oligonucleotides or nucleosides and nucleotides get linked into polynucleotides, right? Sugars, carbohydrates get linked into polysaccharides. You notice the theme here, right? Biology makes almost all of its function by linking together small molecules with reliable chemistry that's programmable, reproducible, scalable, all of that. Okay. So also, and this is Barry's now, I'm channeling Barry Sharpless, okay, many conversations. And also, this kind of attitude, this kind of approach makes modern chemistry, modern life possible. And it all comes from petroleum, right? So we shouldn't be burning petroleum because we pollute the planet, right? The real crime is we shouldn't be burning petroleum because what's in petroleum is irreplaceable to make everything else that we use. So petroleum, for example, makes propane, which gets converted in a catalyst to make propene, and another catalyst makes polypropylene, and that's what this is. Right? Another part of petroleum gets processed into making acrylates, and catalysts make polyacrylates, and that's what these are. right? We're talking about different materials having different functions that support modern life, right? Here's another one. Here's caprolactone. You open caprolactone, you make polycaprolactone, and you go to Scott Hollister's lab, and he prints polycaprolactone into a stent, which gets implanted and saves a kid's life, right? And that polymer comes from petroleum, comes from this attitude, this approach of making big things from small building blocks. Okay, so this is all rattling around in Barry's head, in my head, and all sorts of other things. And then Carolyn Bertozzi is at Berkeley kicking our ass, right? So Carolyn is doing some amazing science here. This is 1997 now, published in Science. A wonderful approach using simple chemistry to take molecules on cellular surfaces that have a unique functional group, here a simple carbon-oxygen double bond, and addressing that, making a bond to that functional group on the cell surface using a particular kind of chemistry. And this is a reaction here that I'm not going to talk about. It was well known for many years. And Carolyn adapted that reaction for use in a biological context. No one had done that before, or not many people had done that before. And certainly with as focused a way to incorporate the desired functional group into the complex biological system in a way where you knew where it was. Okay? So that's what Carolyn's doing. Then a few years later, she seizes on a reaction again that was in the old literature, but she really adapted it. Her group did the fundamental chemistry here to react a phosphine, a molecule with phosphorus in it, with a molecule with this functional group, N3. This is going to appear a lot. It's called an azide. And in this reaction, which you don't have to know, there will not be a quiz, what ends up happening is a bond being made between one of the nitrogens of that azide to a molecule here with this CO group here. And that amide bond is what you're trying to make. And it's on the back end of this phosphine component. So the phosphorus carries one of those components, the azide carries the other component, and you make this bond. Okay, why is this significant? Because again, you can do it in water on a cell without anything else happening. Okay, so Bertozzi writes, right, the phosphine and azide react with each other rapidly in water, room temperature. Both are abiotic and essentially unreactive toward biomolecules inside or on the surface of cells. This is the secret sauce, right? This is the enabling concept. If I can do chemistry in a biological system and only do it where I want to do it, and not wherever else biology presents an opportunity, which is limitless numbers of molecules and complexity, I now have an opportunity to do something selective in a complex system and maybe make a difference in biology. Okay, so this little functional group is the first key. Carolyn, Barry, others recognized that azides were uniquely interesting in their reactivity. Okay, uniquely selective, and we'll talk about that. And then at Scripps, again, we're in the late 90s, early 2000s, lots of stuff is happening. So here's Scripps, right? It's by a golf course, and there's the beach. And this is Richard Lerner. Richard Lerner, I think, could fairly be called the father of click chemistry, because Richard assembled the resources to bring Barry Sharpless there and a number of other people, including myself, and gave us the freedom to do something that was really quite ridiculous at the time. Okay, this is a book that Barry and I read independently that changed our lives. It's a book written by a former editor of Wired magazine. It appeared in the mid-90s, and it's all about evolution. It's about how evolution works, how evolution is the art of knitting things together in a functionally testable way, right? And editing the ones that don't work and coming back and reinforcing the ones that do work. And it really gave both of us a sense that it might be possible to take this concept and run with it. Okay, so this is all bubbling around. And so this is Barry and myself and Valery Fokin, who's an extraordinarily important member of this team, and you'll see why in a minute. And so there we are, noshing around. And this is a creed that Barry lives by right here. Okay? The most fundamental and lasting objective of synthesis is not the production of new compounds, but the production of properties. And so, remembering that biology doesn't get new function by making complex, pretty structures, right? But by knitting things together, Barry came up with the idea of click chemistry. And I put this a little bit out of order, so let me just come over. I'll come back to this. So here was what we called colloquially the click chemistry manifesto, the click manifesto. This is the paper that was really seminal in Barry's award of the 2022 Nobel, because it set forth his vision. It was really entirely his vision on how one can make useful stuff by relying relentlessly on reactions, chemical reactions that are highly reliable. Okay, so some of the headings here might be informative. The process chemistry point of view means process chemistry is once you know what you want, a chemical company has to make a lot of it. So the way you make it the first time is never the way you make it when you're producing tons. The art of process chemistry is figuring out how to produce something at scale. So Barry said, hey, let's dispense with all of the fancy chemistry. Let's do chemistry only that's scalable as a process lab would want to do it. Okay? You need reliable chemistry for that. Olefin-based organic synthesis, that's petroleum making alkenes, which then drive chemistry. We'll talk about that in a minute. Click chemistry in water. Turns out a lot of these reactions work well in water, and so on. This comes from one of the part of the conclusion. Our thinking about carbon-carbon bonds as gifts of nature has been a constant source of inspiration. Okay, in any event, endeavoring to transform these matchless building blocks into substances with diverse and useful functions seems an appropriate way to honor the bestowal of such an embarrassment of riches. And what are we honoring here? We're honoring dinosaurs that made petroleum that we take out of the ground because it has carbon-carbon bonds that we cannot make at scale any other way. Okay, and we take those building blocks and we want to turn them into useful things. And we do that not by making carbon-carbon connections. We rely on nature to have done that. We make connections between carbon and heteroatoms like nitrogen and oxygen. And that's the story of click chemistry. Here's the zeitgeist. Barry Sharpless, Nobel Prize winner, okay, in 1999, tells his research laboratory, which is full of people coming from Germany and India and all over the United States and everywhere else, to do asymmetric catalysis. The man was about to win a Nobel Prize for asymmetric catalysis, turning one-handedness or another, making chemistry that way. And he tells his whole group, we're not doing that anymore. We're going to work on simple reactions. Huh? We're only going to work on reactions that give 100% yield, that work in any solvent, that give no byproducts, and we're going to make stuff using that chemistry. And the people in his lab said, what are you talking about? That's nonsense. That's common sense. Why wouldn't you use chemistry like that? Sharpless's genius, and I will say his fortitude, was to say, oh, no. We're not going to use those reactions when they're convenient. We're going to use them all the time. We're going to be relentless in our focus on making things using only those reactions. The thinking behind that is, if I have a chemical problem that nature has solved with 1 billion years of evolution in a complex structure, that cannot be the only solution to the problem. I have to be able to make a 3-dimensional architecture that serves the same function, and there have to be millions of those answers. And I'm going to therefore find the one that I can make easily, because then everything else is easy. If I do synthesis that way, scale up is easy, modification is easy, right? Adapting it to other things are easy because I know how to make it reliably at scale and I can change it easily. Does that make sense? Right? I'm going to do chemistry like biology does chemistry. Okay, that's Barry's mission. That was his vision. And it took an amazing amount of fortitude because his whole community said, you're nuts. Okay, he was disinvited to give seminars. People would laugh at him openly in seminars when he was talking about this, right? He didn't care, okay, because he knew the power of what he was talking about. And it took me, to my credit, only about an hour walking with him on the beach to become convinced. Okay, it took the rest of the world a little bit longer, but not much longer. And it was only because I had the opportunity to walk with him on the beach. Okay, so this is what click chemistry really is here. From a chemist's point of view, it's taking molecules, building blocks that have a lot of energy built into them, and using those reactions to connect the building blocks together. Now, the reason why this is hard is if I have a lot of energy, I can run off in a lot of different directions, right? So click reactions are ones that have high energy, meaning they're strongly driven, they go downhill, there's a lot of energy built in, but they only do one thing. That combination, lots of energy, but selective function, selective reactivity, that's very rare. And so Barry and I and others combed the literature to find reactions that met that standard, and then we challenged the community, can you invent reactions that also meet that standard, because that really was very difficult to do. So these are the kinds of things, little molecules with that strained rings. We call it spring-loaded synthesis because this little three-membered ring wants to pop open. It's a strained set of bonds. And so if something attacks it, can break this carbon X bond, I now can make a new bond, and I can make it all the time. Okay, no matter what's on the back end of this nucleophile or what's on the back end of this X thing, doesn't matter. As long as they get near each other, I'm going to make a bond. Okay, so the real challenge then, what Sharpless challenged us all was, can we achieve function by using only these reactions as much as possible? All right, so this is the world of click chemistry. I'm not going to take you through it. This is that magic functional group here, these three nitrogens. Okay, and the reason it's magic we'll get into in a minute. Okay, in 1893, the reaction that won Barry Sharpless the Nobel Prize in 2022 was discovered. Okay, this is that paper. They didn't write chemical structures the way we write them now, but what you can see is three nitrogens. Okay, Professor Michael thought it was in a ring. It's actually a line, but that's okay. Plus this thing here, which is really a carbon-carbon triple bond, not written that way. Triple bond plus the three nitrogens makes this ring. Okay, 1893. In the mid-1900s, an absolutely spectacular chemist, right, named Rolf Huisken, a German chemist, popularized this and similar reactions. So this is called the Huisken reaction in homage. And he's still going, he's amazing. But the reaction was very, very slow. And so finally the dam broke with two seminal papers that were honored by the Nobel Prize. This is from Morten Meldal's laboratory. Morten is a peptide chemist at the time. And so he was making peptides and reasoned or figured out that they could put an alkyne on them, react with an azide. And the discovery here was copper as a catalyst for this reaction, because otherwise it's quite slow. And then just a couple of months later, this paper appeared from Barry Sharpless's lab. They developed it completely independently. But again, copper catalysis was the magic ingredient. Okay, I was at Scripps at the time. I wasn't involved in this discovery. That was Valeri here and Barry. Right? But we immediately realized that this is what we needed, because my lab at the time was trying to do this. This is a picture of a virus particle on the left, and we were trying to do organic chemistry with viruses. Nobody had ever done that before. And we were trying to pull the best reactions we could find from the toolbox to make selective bonds on proteins. So we tried everything we could try. We genetically engineered the virus to put sulfur, which is uniquely reactive. We put it in different patterns. We managed to go ahead and put little gold where the sulfurs were. We did chemistry. We were really proud of ourselves. This got into Angewandte Chemie, one of the best journals. I got lovely notes from all over the place. It was great. And we realized we were wrong. Okay? It's not that we were wrong, but our chemistry wasn't nearly as good as it needed to be. Here's a blow up. These little gold things here are actual gold nanoparticles that are attached to this larger protein particle. And you see it's oval, but the gold particle is spherical. Why is it oval? We figured it was just rocking back and forth, and this structural technique kind of captured that motion. But what really was happening was we were getting reactivity where we wanted to, and we were getting reactivity that we didn't want, which was to an adjacent atom, which was sitting there and was reactive in a way that we thought it wouldn't be, because the book told us the reactivity difference was 1,000 to 1 between sulfur and nitrogen in this case, and it turned out not to be true. So the best chemistry known at the time was insufficient to this task. The task being, make bonds exactly where you want on a biomolecule. Okay? There was nothing in the literature that could do that for us until the copper reaction came along. So the reason why this reaction was immediately adopted by scientists all over the world, and that's quite unusual, is for two fundamental reasons. Number one, the reactive groups are completely inert, almost completely, pretty much inert to everything else. They don't have any intermolecular interactions. For the chemists in the audience, they don't do hydrogen bonding. They're not acidic. They're not basic. I can't deprotonate them. They're completely unreactive. And they're small, right? Little 2 atom, 3 atoms. So they don't change the properties of what you attach them to. So I have an invisible handle. And the other thing that they're good for is they're easy to put on stuff. Synthetic chemists love these things. They're really easy to install. So I can put them wherever I want them. They won't change the properties. They'll hide out until I present them with their reactive partner and a catalyst, and then I can make a connection. Okay, the truly unusual thing for this reaction, and this is not true for any other reaction that we know about so far, is that the downhillness, 50 kilocalories per mole downhill. In other words, there's a lot of energy built into these things, but it's very slow. That combination is entirely unknown other than this, to this extent, other than these reactive groups. In other words, imagine you're sitting on the top of a mountain, you got a race car, you're going to push off the top of the mountain, but all around you is a hill you got to get over when you're sitting in the caldera of the volcano, right? I can't get this thing over the hill to race down the mountain. So getting it over the hill to race down is the province of a catalyst. And if I can do that, I have enormous power at my disposal to bond things to each other. Okay, so that's the whole secret of click chemistry. That's why click chemistry was honored in this way. These are some of our papers after the discovery of the reaction. This is all our work, and there are others. And we did a bunch with this reaction because we immediately found it useful. The first thing is we used it. Okay, so this is the first paper on bioconjugation using click chemistry, using the copper reaction. And here's just a sequence. Here's a virus particle. We put 60 azides or alkynes on it, and we made a bond happen. And this is the line in the table that shows you, hey, we got 60 attachments, we got 100% loading, we got 96% yield of this structure out, and nobody could do this before. Why? Because if I have 99% yield on each of those 60 reactions, I still only get 50% yield of the product. Right? I need 99.9% yield for each of those reactions. And there was not a reaction known before the copper reaction that could do that on a protein. Right? So that was lovely. We then figured out how to put these unnatural amino acids with these reactive functional groups, these invisibly reactive functional groups, we could code them in genetically. So we figured out how to do that so we can put them exactly where we wanted them on our structures. And so then we started going to town. Right? These are pictures of viruses, and here's a cartoon. We put polymers on these viruses. We wrapped them up in organic spinach and made them very, very stable so we could boil them and they would be fine. And we could administer them to cells and they would do something else. And we just kept going. Here's another image from one of our papers where the reactivity was predictable. What we're looking at here is putting different kinds of sugars on this common platform. These are very different sizes, but we could program the connectivity because it all was the same. It's just this little azide at one end and the alkyne at the other end, and we could dial in exactly how many we wanted, no matter what it was. And so we could make things that were unable to be made before to ask questions of biology or influence biology. All right? More of this happens all the time. Okay, here's Carolyn Bertozzi now, not using copper because copper is toxic to cells. So we and others have worked on doing the copper reaction in the presence of cells, and we're not there yet. We can do some of it, but not a lot of it. Anyway, Carolyn says, let's use this Staudinger reaction, and now I'm going to allow biology to put in an azide, and I'm going to be able to program cells by changing their surfaces using this orthogonal chemistry. That's bio-orthogonality, doing reactions in biology where you want and not where you don't want. Okay, that's that term. And then Carolyn said, okay, this ligation reaction, this Staudinger reaction that I had talked about before, wasn't quite good enough. So she went back to the old literature, more German literature, 1961, looking at the reaction of an alkyne, in this case with this kind of molecule, doing a reaction that made new bonds. The inspiration here was this alkyne in this ring was unusually reactive. So Carolyn said, okay, we're going to try this now with azides, and it works. And that's now copper-free click chemistry. Okay, these reaction is basically the same, an alkyne with an azide. Now you don't need copper to do this. And Carolyn's lab, and again, countless other laboratories over the world use this. The reason why one might want to use this reaction, this guy here, this little alkyne and an azide here, or this reaction that Bertozzi developed, just depends on how big a molecule I can tolerate, right? This version, this copper-free version, requires strain, requires a ring for that alkyne to be reactive. And that gives me a lot more atoms I need to build in. So very often, this kind of molecule, if I attach it to something, will change the properties of what I attach it to. The little 2-carbon alkyne won't. And so in general, we prefer the 2-carbon alkyne when we want to preserve properties and make connections. When we don't care, then we prefer this chemistry because it's easier. Right? All right. So I'm now just going to race through some other stuff that my lab did at the time just because it's fun. So we knew copper was a catalyst. We knew azides and alkynes made triazoles. We knew this 5-member triazole ring actually binds metals. And so we were the first ones to publish material science using this reaction. And what we did was adhesives. In fact, if you take these kinds of azides and alkynes and put them between 2 copper plates and you wait a while, you're going to make something that, I'm going to skip the data, that sticks and you can support a graduate student with 10 milligrams of adhesive sitting right there between 2 copper plates. Okay, so you make really strong adhesives because this reaction catalyzes its own production because I have copper from the surface of the metal leaching in and making more catalyst, right? It was a lot of fun. Here's a case where we glue the nail, this copper nail, to a copper plate, and you push on it with this thing to see how much force you need to snap it off. How you judge the strength of the adhesive. Well, this adhesive was so good it just bent the nail. Right? So we had a lot of fun with that. We also did most of the work on how this reaction actually happens. And this is, in a career of looking at the mechanisms of reactions, this is the most complex reaction that I'd ever dealt with. And I'm not going to take you through it, but we figured out how it works, and it works in different ways in different environments. So we reported to the world that you need 2 copper ions to make this reaction happen, that when you have something binding the copper catalyst, the mechanism changes. Again, I'm just going to skip through all this stuff here. Oh, here's an important note. The rate constant here goes up by a factor of 10 to the 8 relative to the uncatalyzed reaction. So azides and alkynes, when they get together, if you have them in solution, they will not react. Half-life of 40 years, okay, for that reaction. You put copper in, you can get it to happen in a couple of minutes. Okay, figuring out how that happens was one of my proudest achievements in figuring all this out. And so very complex, but we all worked it out. The literature uses this. This is one of the neatest slides for you chemists. This mechanism responds to its environment. Okay, here's the true catalytic cycle. 2 copper ions are held together. One of them binds the alkyne. The other one binds the azide, bringing them together in a particular way to make that 5-membered ring. And then that copper-carbon bond has to be disconnected to make the final product. That's the catalytic cycle. Okay? The interesting thing is this ensemble of elements breaks up and reassembles if the environment challenges it by binding to copper in a different way. It is a remarkably slippery catalyst, and therefore it works everywhere. It just has to find some opportunity for the coppers to get together in this way for just a brief amount of time, and the reaction can happen. Okay, so this paper where we synthesized all of that and advised the world on how to do this reaction using biological molecules. So this paper has now really helped the field go forward using the azide and alkyne in biology. Okay, I want to tell you one more conceptual story. Remember, we have 2 functional groups, azide and alkyne, that do not react with each other until you bring a catalyst to play. The other way you can get a slow reaction to happen is just hold them together for a long time. Okay, so if I'm reluctant to react with you, right, to engage with you, if you force me to stand next to you, I'm going to talk to you eventually, right? So it's the same principle. And the lovely part about having reactants that don't do anything else is it doesn't matter how fast or slow that chemistry is. When a connection is made, you know that the things have been next to each other. They won't make a connection until they are next to each other. And for a lot of these, they have to be next to each other for a long time. Why is this useful? Well, if I want to make a drug and I want to hit an enzyme and I don't know what the structure of the enzyme is, I typically want to bind to that enzyme. I want to find a molecule that grabs onto that enzyme. And if I don't know what the structure is, I can't design anything. So instead, let me take combinatorial chemistry, make a bunch of molecules and put an azide on them, and take another bunch of molecules that I think might bind to the enzyme and put an alkyne on them, and just mix them together. Here, X and Y are 2 functional groups that might or might not react. Right? They'll only react if they're next to each other. So now I get a bunch of molecules, they come in and out, on and off the protein, and only when they're held together for a sufficient amount of time does X and Y get together to make a bond. If X and Y are really slow at this, the only time you get a bond is when the pieces are there for a long time. And the only reason they're there for a long time is if they're pretty good binders. So I have a way now, without knowing anything about the target, to play Battleship. You guys have played Battleship, that game, right? I can play molecular Battleship and find two things that hit the target and that connect to each other. So I now have made a molecule that binds it much better, right? If I bind with my right hand, I can rip that off. If I bind with my left hand, I can rip that off. But if I bind with both hands at the same time, I'm holding on a lot tighter, right? It's a lot harder to get me to separate. That's the concept. Okay, so this click chemistry is not done with copper. It's in fact designed to be slow, and it's azides and alkynes. And here was our initial target. So this is the enzyme acetylcholinesterase. It's vital in your brain if you have a malfunction of acetylcholinesterase. If you get snake venom toxin, or if a snake bites you, it stops this enzyme from working. So you have seizures and you die. It's intimately connected with dementia. So this is a really interesting target. It happens to have this little gorge, actually quite a deep gorge in it, and we could fill that gorge up. And so in 2002, we published this paper in which we allowed the slow click reaction to connect two pieces that we knew bound in the enzyme pocket. We cheated because we knew what those were to begin with. But we made a family of molecules with these little connectors and allowed it to assemble by itself in the molecule. And what we got was an unprecedentedly tight binding molecule to this structure, even more tightly binding than snake venom toxin. Okay? And it was because we allowed the molecules to tell us what was bound and what could reach each other and sit there for a long enough time. So this in situ click chemistry, as we call it, and I'll skip this. Here's a picture of the gorge, and the protein changes conformation, which was unusual. So this has now been picked up and used by a number of laboratories. Here's a lovely one from Jim Heath, who was at Caltech at the time, taking peptides, having them bind to the surface of a protein, and using this selective reaction to click them together. Here's another one. So this is one of the molecules they made. Here's another one from Matt Disney's lab in Florida, making now a molecule inside a cell for muscular dystrophy from two parts that each one alone does not affect the function of the cell, but when you click them together, you have an active drug. So here you're making the drug inside the cell, right? Again, using the ability to bind and this reaction to happen only when the pieces are next to each other. So this is why click chemistry is useful, all sorts of stuff. Here was another reaction, yet another addition to the click chemistry family that I helped Barry develop years later. I'm going to skip it because it has the same attributes: sluggish reactivity until we turn it on, either with a catalyst or by proximity. What power that is to interrogate complex systems, right? So I'll skip through all of these. Oh, I should say, this was our favorite, absolute favorite journal cover character. They wouldn't let us use this because apparently the Superman image is copyright protected. So we could not get this on the journal cover. I was so disappointed. But anyway, so we're going to skip all this. Okay, uses of click chemistry. So here are citations for click chemistry from 2002, right, the year after it was published, and we're looking at 700-800 publications citing this one paper each year, which means that I simply cannot tell you all of the ways, or even a fraction of the ways in which this reaction is used. But I hope you can appreciate why it's so useful. It takes chemical synthesis out of the hands of the artistes. We still need the artistes, right? We've got to make complex molecules because we got to make good building blocks, and we sometimes can't rely on this approach. We still need them, right? But it allows you, if you have access to building blocks, to making your own stuff. It's a democratization of chemical synthesis, but more importantly, it allows you to focus on why you're doing it, not how you're doing it. Why am I making something? For function. Click chemistry relentlessly drives you to function. Why? Because you get no credit for doing a click reaction. Anybody can do it, right? So fine, let's do it. Let's forget about how to do it. Let's do something important. Okay, that's the whole magic here. Okay, so I can't resist showing you just a little snippet of what we do now these days with click chemistry. So here's a dialkyne. You can recognize that. Here is, I don't even show, here are the diazides. If I have alkynes and azides clicking together, right, I got two and I've got two, I'm going to make a long chain. right? So this is a new family of polymers made by this reaction. What good is this family? This family separates the components of crude oil. Okay? Why do we want to separate the components of crude oil? And it makes a membrane. So this is what we're doing with the wonderful group of Ryan Lively. The reason why we want to do this is because it costs 1% of the global energy use by humans on the planet to separate the components of crude oil. 1% of all the energy we use. Okay, 10% of all industrial energy is separated in components of crude oil. Okay, why? Because crude oil is tremendously useful, right? We should not burn it, but we still need to separate it, right? And so these membranes are unique in the ability to take crude oil and separate out the low molecular weight stuff. Here's another one. Crude oil, low molecular weight stuff, just by pushing it through a membrane rather than distilling it, which takes a lot more energy, right? How did we get here? Right? I didn't come to Georgia Tech knowing what membrane science was. Right? I came to Georgia Tech because there were people here who knew what membrane science was and all sorts of other things. Right? But the reason again is because I could gain entry because I have a powerful reaction at my disposal. Right? And to be honest, Ryan Lively doesn't need me. Okay? He could do this chemistry too because it's easy. He actually needs this because the building blocks, are too difficult for his lab to make. Okay? But the point is, we got into this field because we had powerful chemistry at our disposal. Okay. Here's another thing. We work a lot on immunology, right? Totally out of the box thing. We work on immunology of carbohydrates. Why? Because if you can recognize a disease-relevant carbohydrate, you have a new vaccine. Okay. And carbohydrates are amazingly complicated, difficult to recognize. But with the magic of click chemistry, again, we don't have to worry about how we make something. We can make this piece, which is hard enough, or have collaborators make it for us. But once we put a little azide on it, we can attach it, make a vaccine. In this case, we're putting it on viruses again. We make vaccines because viruses are immunogenic. And we have the first general method with collaborators at Scripps to make high affinity antibodies to carbohydrates generally. Nobody else in the world has been able to do this before we got into this because of click chemistry, right? Because we had the opportunity to do this because we didn't have to worry about how we assemble these structures. Okay, and here's just our final story, which I want to make sure to tell because it's an immunology story and it's relevant to children. So leishmaniasis is a disease coming to the United States. Of course, it affects large swaths of the world, primarily where the insect vector. So it is an orphan disease, right? It affects millions of people worldwide. It's a parasitic disease and can kill you or can cause skin lesions, cutaneous or visceral leishmaniasis. And it's coming north because climate change. Right, because the insect vector is coming north. And we're very interested in immunology in my lab. And it turns out, again, through amazing people, that we realized that this carbohydrate was key. If we could raise an immune response to this carbohydrate, maybe we would have a vaccine against the organism, the Leishmania parasite. And so we did click chemistry again, azides, alkynes, put them on particles, make a negative control, immunize mice, and we protect mice. Okay, these mice haven't been immunized. They get the disease. These mice have been immunized. They do not get the disease. We have a viable vaccine candidate. Okay, lots and lots and lots of work go into that. But again, we would never get into this if we didn't have the powerful chemistry. Lastly, I lied. This is the last story. Here's fentanyl, right? An epidemic by any measure. Okay, as it turns out, and again, I'm racing through this, there are many derivatives of fentanyl. So fentanyl is a problem because it's 800 times more potent than morphine, right? 30 times more potent than heroin. Okay, so it's a real problem, as you know. Many, many, many deaths each year from fentanyl overdose. This is the fentanyl structure. It's ridiculously simple. And because it's ridiculously simple, there are more than 100 variants of fentanyl that people have made and are known circulating or being used. And many of them have deleterious effects, but some of them have the same effect as the parent fentanyl. And with our friends at the CDC, we identified 73 different derivatives. The CDC, with our help, tested every commercial kit that was known to detect fentanyl with the idea of how well do they detect these fentanyl derivatives. Okay, nobody had done this before. The problem to be solved here is a law enforcement officer arrives at a scene with a white powder, and they don't know what it is, and if you breathe fentanyl, you die. Right? So they need to know what it is right then and there. They can't send it off to the lab. And if the white powder is a derivative of fentanyl, anything above a purple bar here, any of these guys, like from up here on up, and you have a commercial kit on site, it will not tell you that it's a fentanyl derivative. So this is a real problem to be solved. We turn to the immune system to solve it, but again, empowered by click chemistry, where we could make variations of fentanyl, put an alkyne on them, attach them to our immunogenic platform, and allow the immune system to make binders of each of these derivatives of fentanyl. Okay, so we did this, a rapid attack on the problem. The mice did most of the work. Okay, we make the reagent, we put it in mice, we get immune responses that are amazingly good. We were all shocked. And we generated antibodies, again, with the CDC as an intimate collaborator. We generated antibodies where before we could detect only like the top quartile. Now we can detect anything that has a blue square, which means these are antibodies. So if you can find an antibody that detects with blue, we can detect that fentanyl derivative with high sensitivity. There's a class of fentanyl derivatives we cannot detect for reasons they're just too small, but most of them we can. So the next steps here, getting these into field kits, making potentially vaccines, again, a completely different area of endeavor enabled by click chemistry. So that's the kinds of stuff that happens, right, when you have a discovery of this magnitude. So I really want to, of course, thank these people, right? Whoops, come back here. Okay, I want to thank Barry, of course, particularly. We have a long and incredibly, I'm just incredibly fortunate to have the relationship that I've had with him over many years. Caroline's been an inspiration and a friend, and just her chemistry is just, we look to all the time. And Morton, really a remarkably insightful scientist. Again, I didn't have a chance to talk about his work, but his laboratory, of course, was key here, and he's also a friend of long standing. Lessons, perhaps, I think I have only one or two here. Really, first and foremost, really is gratitude. I'm thankful to those people. I'm mostly thankful to these people who were, this is most of my group here, who have decided to go along with me on this ride and use click chemistry for all of these purposes. We don't use click chemistry just to use it. We use it because it gets us where we want to go, right? Remember, we get no credit for doing a click reaction, right? We only get credit for making a vaccine. And that's what we want. And so I also want to thank, of course, the sponsors. Much of this work has been funded by the NIH, a little bit by NSF. We've had really wonderful partnership with Pfizer over many years. ExxonMobil, I alluded to before, they didn't get the credit they deserved for the crude oil separation work, really are great partners. And I'm immensely indebted to Children's Healthcare of Atlanta, who has provided both a rich area of endeavor for me, working with children's, working with the people who work with children's, trying to get people to solve problems for pediatric health. And I'm immensely indebted to Jimmy and Helen Carlos for providing the resources for the chair that supports this work and a lot of what we do. Remember I said Richard Lerner was the father of click chemistry? And he really was, because he had the vision, he also had some really amazing ideas, but he had the vision to bring people together, give them resources, and let them go, and let them run. And Jimmy and Helen, thank you, because you provide that kind of flexibility for us and that kind of freedom, and it really is quite rare. And I'm really immensely grateful to the Georgia Tech community. I came to Georgia Tech from Scripps because I couldn't do material science at Scripps. And Georgia Tech is the Scripps of material science. And it also turns out that Georgia Tech is among a community that is equal to Scripps in immunology. The Atlanta immunology community is amazingly good, and we have wonderful people here who contribute fundamentally and crucially to that. And so immunology, I'm very happy here. And in every respect, doing chemistry, doing science, integrating in this marvelous, diverse community of scientific interests and of people with different career goals and all of that, I just couldn't be happier. And so I'm really grateful to all of you, and I'm grateful mostly to this group, and thank you for your attention.
Wonderful. Thank you, MG. We have time for a few questions if we have them. I want to say there's 140 people online for the talk.
Jeff. Oh, yeah.
Beautiful talk.
Thank you.
So having elucidated how simple the chemistry is, in quotes, yet the applications are profound, what's next?
I have this all queued up because Barry and Hartmouth and I wrote a piece for Nature Synthesis for their first issue earlier this year. And it's kind of a retrospective and it's a little forward-looking piece on the future of click chemistry. So you can read that. Just look up Nature Synthesis in my name and you'll find it. But what we really hit on was landing even more firmly on this idea of proximity-guided synthesis, and taking it another step further. And that is to try to find chemistry that not only relies on where you are in terms of physical location relative to something else, but also what the environment is, what the chemical environment is. Can we tune reactivity in a complex enough way so that not only can I hit every serine protease, let's say, which is the business end of activity-based protein profiling, for example, but not only can I hit every serine protease, but now I want to selectively hit a serine protease that is doing something in a neuron. Okay, that's a challenge, right? Or I want to hit a particular isotype of a particular enzyme versus another one. Very subtle differences in structure, in environment. The reason why Barry and I think that this is possible is that the idea of proximity plus connectivity gives you more molecular information to read out. Not just proximity, but what is the dipole moment around the environment, right? Is there a hydrogen bond there available or not? I have more molecular information to influence the rate constant of the joining reaction. That, for me, is where click chemistry might be going in the future. The other way it's going is really trying to do evolution the way that biology does it, but bringing in click chemistry in the evolutionary cycle, right? Can I bring in reactants and in a separate step from the biology, can I induce a reaction to happen outside of the cell and then feed it back to the cell to have it do the cycle some more? Right. So that in my group years ago was called the Crazy Project. Okay, and it never worked, but we'll keep trying.
MG, what was special about the copper catalyst that other metals didn't have?
Yeah, so copper is unique. It has two things that are really important. Number one, it's a smooth ball. It doesn't, so copper-1, in the copper-1 oxidation state, it's a D10 metal, which means it has no particular orbital orientation that it favors. So you look at most metals and they have rigid coordination environments. Copper doesn't care. So it's very slippery. So it will bind, it will leave and bind to various things. The world expert in copper chemistry is sitting behind you. That's Christoph Farney in copper and biology. And so Christoph knows far more than I do, but essentially copper is a smooth ball that engages in the kind of bonding, in a multiplex kind of binding, so it can interact with a variety of different things, but it's not rigid. That's number one. Number two is it has just the right degree of electron richness so as to provide enough electron density to make this reaction go. We know from our studies, the more reducing copper is, the more electron rich it is, the faster the reaction. However, the more electron rich it is, the faster it reacts with oxygen and gets killed. So we try to find a balance, and we were able to dial that in, but that's the magic of copper. Slippery as hell, okay, but bonding enough with these kinds of reagents to make the reaction happen. No other metal does it.
Hey, MG, I want to make an observation and ask you to comment on it, and it's really directed towards the young scientists who are here. The vast majority of the papers that you showed were published in really good but very accessible journals. JACS, Angewandte, JOC. You saw very, very few papers in Science or Nature. So I think a comment that I would make is that if you do really good work, you get it out quickly in a journal that's widely read, you're going to get credit for it. And sometimes it's not necessary to spend the 1 or 2 years in the gymnastics necessary to publish in Science or Nature. So just something I took away from this is if you just do really good work, get it out there where people will read it, you're going to get the rewards. Yep. Do you agree with that? Can you comment on that? Because you also publish in Science and Nature.
Well, I can go farther than that, and I'll just relate a story from Barry. So Barry's first Nobel was an asymmetric epoxidation, right? And that, not the first paper, but the second paper, which really nailed it. That was rejected from JACS, okay, and got published in JOC, in Journal of Organic Chemistry, right? And Barry always said, if it gets rejected, I know I'm onto something. Okay? And you know that we're not all Barry Sharpless, but the lesson that you're trying to convey here is absolutely true, right? It's got to be good work, right? But you know, good work in a good journal, that's fine. I totally subscribe to that theory.
take a quick one here, and Cheng has the last one. You're me first.
All right. Thank you, MJ. This is fantastic talk. So I want to take you out of biology into process, right? You said one of the philosophy is be able to do this in a scale that process chemists want to engage in doing chemistry. So it's turning out now that triazole is appearing in drugs, right?
Triazole is?
They appear in drugs,
yes,
and
yes.
we're using click chemistry to make them. In fact, I mean, I believe there are 2 or so drugs now that are in the market, right? But one of the components of click chemistry has a bad reputation, and that is azide. Mm-hmm. In terms of whether you can make it in large quantities and be able to keep it safe for process chemistry. So can you comment about what is known about the safety profile of triazole, right, with regards to being amenable to process chemistry?
So I'll just reiterate for those who didn't catch. So the azide functional group has a bad reputation as being explosive. It is high energy. Remember, I mentioned there's a lot of energy that we use in that reaction. So again, I'll tell you a Barry story, perhaps. Barry was always attracted to these high energy things, in part because there's reactivity there, but also, frankly, because it keeps the riffraff out. In other words, if you can do that chemistry and do it well and safely, you're not going to have a lot of competition, right? So now that's a little bit flip, but we paid great attention in the early days to azide safety. We still do, right? The azide and we have robust ways to worry about this, but it turns out azides are not that bad. There's a class of azides that are much more explosive than others, but it really isn't. There are companies out there that will make azides for you on ton scale. They're the same companies that do diazomethane chemistry at ton scale, by the way, right? So you can do a lot worse things at large scale than make azides. It used to be in your car, you had a pound of sodium azide sitting right in front of you because that was the thing that made for airbags. Right? So if you detonate sodium azide, you make nitrogen gas and you fill the airbag. They don't do that anymore. But if you have a car that's older than maybe 2005, you got a pound of azide sitting right in front of you. No.
MG, this is wonderful talk. I got a lot out of it. Thank you very much. So I want to take back to what this gentleman just said and maybe go a step further. So, one is the publication where you publish. The other is you said that this person, I forgot his name, in Script that brought Barry into Script and just provide them resource and let them go. I don't think the current system at Georgia Tech allowed that. How do you get tenure, right?
I keep asking Xiao, and he keeps saying,
you are department chairman.
We're not.
So what does it say?
Sorry, the question was how do you get those resources, or how do you get tenure?
I mean, the current thing, they put you there and they kind of, you're a male, cash cow, generate grant, generate paper. It's not like creating creativity, promoting creativity. It's squeezing people productivity. So,
This is a longer conversation, obviously. But let me say that, and those of you who are experienced enough or old enough to have sat on many review panels. Mohan, you have your hand up.
No, I just have a comment for this question.
Yeah.
The previous Dean of Sciences, Paul Bovart, now that he's not here, I can quote him now, but by all means, he will come and kill me. Said, Mohan, do you know why we are like this? Men and women with spreadsheets are going to ruin us. That was his precisely, this is what happened at the laboratories. The labs does not exist. It had 12 Nobel Prizes. There's Nobel labs. It will never happen.
I am a great fan, however, of peer-reviewed science, having sat on many NIH study sections, many NSF panels. I will tell you that, I mean, as you know this, Cheng, of course, it's a really great process, right? The science that you see and you hear about and you have to get through that kind of critique is phenomenally useful. So there are not many Barry Sharplesses in the world, right? But Barry was able to do what he could do because he also had the ability to go the normal route. It wasn't just all Richard Lerner saying, okay, here's a pot of money. It was Barry being able to publish papers and do creative science to attract the people that he needed to attract. And Richard gave him the freedom to not worry about a grant for click chemistry for a while, right? But eventually he had to get one. And eventually, you know, Barry didn't write his grants, but that's okay. But eventually, it proved out good science gets paid in our system. It takes a while, and for young scientists, it often takes too long, right? But you sit on enough study sections, good science gets paid. So keep at it, and you will get the resources you need. Our research budget, non-defense research budget in the U.S. is $80 billion. It should be $500 billion, right? But $80 billion goes a long way, and there's enough out there to support the folks who come from Georgia Tech and do great work.
Great. Thank you. So with that, please join me in thanking MG for a wonderful